Assalamu alaikum. I'm Professor Dr. Haider Jawad Mubarak. This is a presentation to the topic of comparative anatomy of the urogenital system and part of the curriculum of medical biology for the first class, second semester, College of Medicine, and Nahrain University. The kidney in higher vertebrates, including human, structurally is formed of cortex to outside and a medulla to inside, which is formed of many pyramidal shaped structures. This is the gross macroscopic structure of the kidney of higher vertebrates, including the human. Macroscopically, you may find that the kidney is formed of three structures mainly. The first structure, which is called the renal corpuscle, it is formed of a tuft of glomerular capillary surrounded by a capsule of membrane, which is called Bowman membrane or Bowman capsule. This is the first part of the histological or microscopic structure of the kidney. The tuft of glomerular capillary filters the urine into the capsule into the Bowman capsule. And then, after the renal corpuscles, the urine passes to a series of tubules, which are proximal convoluted tubules and distal convoluted tubules, that regulates the urine filtrated from the renal corpuscles. And after that, the urine passes to a collecting duct that also filters or regulates filtration of the urine and collected it to be excreted out by the ureter to the renal bladder and so on. Actually, some of these structures are located in the cortex and the others are located in the medulla of the kidney, and I think this figure is so clearly showing that the renal corpuscles, including the glomerular tuft of capillary and the Bowman capsule, in addition to proximal and distal convoluted tubule and part of the loop of Henle, are located in the cortex. The other parts of loop of Henle and the collecting ducts are located in the medulla. Such a structure of the kidney that is seen in higher vertebrates is embryologically derived from three kidney systems. Each of these systems is located in a position inside the body of the embryo. So they are classified as three kidney systems inside the body of the embryo, depending on the position of each of these three systems. The systems include, number one, pronephros, which is the kidney system that is developed in the neck of the embryo, in the cervical region of the embryo. The other system is the mesonephros that develops in the thoracic and upper lumbar region of the embryo. And the third system is the metanephros that develops caudal to the mesonephros system, which means in the lumbar, in the lower lumbar region of the embryo and sacral region. So you can imagine that these three systems forming the kidney of embryo simulates the structure of the kidney of the earlier vertebrates, which is called archinephros. As this kidney of the earlier vertebrates, the archinephros, extends along the whole body of the vertebrates from the neck down to the sacral region. And this is a figure that shows you the structure of the kidney in lower vertebrates or in earlier vertebrates. It extends along the whole distance from the neck to the thorax to the abdomen and down to the sacral region. And what's going on that during evolution, the higher in developments of vertebrates shows segmentation of the kidney to be derived from part of these three systems. For example, the pronephros generally is found only during embryonic life. So the pronephros in vertebrates is a temporary kidney system. It is seen in earlier embryogenesis, but it disappears in later stages of embryonic development of vertebrates, of course. While the mesonephros may form a kidney system in the embryo and in adult, in fish and in amphibia. While in amniotes, which are vertebrates having amniotic sac in their embryo, including the human and mammals, the mesonephros seen only during embryonic life. It develops for a short time and then it will disappear in adulthood. Therefore, in amniotes, including mammal, human, the kidney is derived or are derived from the metanephros, the last kidney system that is located in the lower lumbar region and sacral region. Therefore, in amniotes, mammals and other amniotes, the kidney develops in the lower lumbar and sacral region inside the pelvis. And then, during development, the kidney that develops in the pelvis ascends higher and higher to reach its final position into the abdomen. Because of this embryological origin that is described for the kidney system, in a human, you may find abnormalities just like these. More than one kidney, as it is shown in this figure. And such a duplication of the kidney is very easy understood in terms of the renal system that I'm just describing. If the kidney is derived normally from the metanephros, it will be one kidney, but if it will develop from one kidney system, maybe you will have more than a kidney and C condition. Abnormalities in the renal system therefore is related to the origin or the precursor of the kidney systems during embryonic time, and as you know, embryogenesis recapitulate phylogenesis. In regard to blood supply to the kidney system, if you look to the figure of the early vertebrate kidney, the one that we call it archinephros, you can see that the aorta, the abdominal aorta, could give a branch to each of the kidney system. And therefore, each of the renal systems, the pronephros, the mesonephros, and the metanephros, having a blood supply from the aorta. Therefore, you may find that some vertebrates may have more than one blood supply to its kidney, indicating multiple origin of the kidney from the parts or from the segments of the renal system. For example, In birds and reptile, the kidneys shows 2 or more renal arteries, while in mammals, including the human, the kidney is normally having only 1 renal artery. In consideration to urinary bladder, in tetrapod, the urinary bladder is seen and it is functioning for excretion of urine when it is desired, not when it is produced. However, also there is variability. There are many variabilities that could be seen, including that some vertebrates, just like the Ignatians and the snakes, crocodile, lizards, and bears, do not have urinary bladder. They have a cloaca, and other vertebrates having urinary bladder, just like the amphibia or the mammals. And other vertebrates, which are the amphibia and mammals, the bladder is derived from the cloaca, of course. That is, in summary, what is described for the